Hello and welcome back, everyone. It is my great pleasure to introduce our first keynote speaker of the Virtual Global Summit, Dr. Evan Eichler. He is a professor of genome sciences at the University of Washington and Howard Hughes medical investigator. Evan has been a champion of genetic variation, in particular structure variation, for many decades now. And he's also been a champion of the PacBio technology for a long time now. And so we're really pleased and grateful that Evan will give the keynote presentation at the Global Summit. And with that, I will pass it over to Evan. I should mention that you can submit your questions during Evan's presentation. Please do so. We'll collect them, and then we'll have a live Q&A session after the end of Evan's live presentation. With that, I'll pass it over to Evan.
All right. Thank you, Jonas, for that introduction. I'm going to share my screen right now so we can get going on this right away. Let me just adjust this. So it's a pleasure to be here, even though it's only virtual, to share with you some really recent advances that have happened in our lab and actually outside of our lab as well over the last, I would say, last 2 years. Maybe just to kind of as an introduction, I think one of the things that ties everybody that's probably listening in on this conference is that as geneticists, we are fundamentally interested in understanding really the full spectrum of genetic variation. And that has been problematic, I would argue, for the last 20 years, really for 2 reasons. Let me just reset this. One is that genomes are complex in their organization. What I'm showing you here is one form of complexity with these blue lines indicating large, highly identical duplications that exist at multiple locations. And I'm showing you here the pattern of intrachromosomal duplications contrasted with that of interchromosomal duplications. So about 5% of our genetic code is made up of such complicated, large, highly identical duplicated sequences. There's close to 1,000 genes embedded in these regions. And in addition to this complexity, the one thing I didn't highlight here is these purple bars. These purple bars represent areas of the centromere, Aerocentric regions that 20 years after the Human Genome Project was declared nearly finished have still not been resolved. And I hope to dispel you of that notion today. The second major problem, which I think is important, is that the regions that I just showed you are particularly enriched in copy number variation. This is just one view showing chromosome 16, and a heat map that has been generated from the sequencing of about 100 human and great ape genomes. And on the right-hand side, you can see all the copy number variation estimates. Wherever you see black, that means that it's a unique sequence. But the really important point I want to convey is that for years, we've recognized that there is, in fact, extensive copy number variation in these regions of the genome, both within and between species. So for example, you can see a human-specific duplication of hundreds of kilobases located here and not present in any of the other apes that are shown below. We know there's 3 times the amount of variation when compared to single nucleotide variation represented at the base pair level. But important for this discussion today is that 50% of all normal copy number variation maps roughly to this 4 or 5% of the human genome that I showed you before. So by not sequence resolving this type of variation, or these regions, we are also not understanding a big chunk of the genetic variation that actually contributes to us as a species, but also contributes to disease within our population. So the problems are really quite simple. There are complex regions of the genome that are difficult to sequence resolve, and these are enriched for structural variation, and particularly unresolved genetic variation. Short read methods have been great, so such as Illumina, for inferring where that variation is, but not sequence resolving it. And in fact, if you look at your favorite Illumina genome that's been mapped to the human reference, these regions typically receive very low map Q values, and so they're typically thrown out from subsequent analyses. So if we have disproportionate amount of variation mapping to the regions that are not being assayed, I think it follows that if there is any disease associations or any significance with respect to whether it's gene discovery or whether it has to do with essentially the status of methylation across regions, these regions are woefully underestimated. So I would argue that this problem is now becoming solved, and it's being solved from what I think is called a long-read genome sequencing revolution. I know we've all heard the term revolution in the genomics field probably far too often, but I believe this is a reality with respect to what we're trying to do, at least, which is to characterize these more complex regions. So both PacBio with their continuous long-read sequencing and circular consensus sequencing, as well as Oxford Nanopore with essentially their long reads that they're capable of generating with both the GridION and to a lesser extent with the PromethION system. I want to highlight two advances in, I would say, the last 2 years, which I think are particularly exciting. One has been really the evolution of high-fi PacBio sequencing. So this you'll hear much more about, I'm sure, many times today, but this idea that you can now get long reads that are highly accurate. And the way this is achieved by actually having the polymerase go around using the SmartBell structures, go multiple times around essentially a sequencing insert to generate essentially a consensus sequence, which is highly accurate, greater than 99.9% accurate. And the other, which I'll show you as well, is the use of particularly ultra long reads. So I will define ultra long reads as reads greater than 100 kilobases in size. And how these two technologies together, I think, have actually solved many of the most complex regions of our genome. So my objectives today are really threefold, well, four if you count the applications. I want to share with you kind of a little bit of a history in terms of how we got into sequencing, and particularly from the purpose of resolving at the sequence level structural variants. I then want to move to the telomere-to-telomere assembly of chromosomes, work we've been doing particularly with Adam Philippi and Karen Miga. I then want to talk about some more recent work that we're doing with Tobias Marshall and the Human Genome Structural Variation Project to actually phase and sequence assembly multiple human genomes. And then I want to end with, we have time, with the different applications with respect to variant discovery, disease, and evolution. So in the beginning, I think this is now over 7 years ago, Jonas, when we, maybe even longer than that, when we first got involved in this, we were interested in actually sequencing a human genome from one very specific perspective, and that was to characterize structural variations. So these are insertions and deletions and duplications greater than 50 base pairs in size. And so at that time, sequencing a genome was expensive. It was over $70,000 to generate the data at that time. And so we decided that we would focus on a genome where we would have a slight advantage. And so we focused on specifically hydatidiform moles. For those of you who don't know what a hydatidiform mole is, this is essentially an aberration of fertilization in which an egg is essentially loses its maternal complement, and such that the monospermic DNA is the only thing that remains, essentially eliminating all allelic variation, and therefore simplifying the ability to discover structural variation at different repetitive regions as opposed to allelic variation. So this is the work when Mark Chasen was in my group. Very simplistically, we took the long reads, mapped them back to the human reference, looked for signatures that were indicative of structural variants. Then when we identified those signatures, recruited those reads, did local assembly, and resolved the structural variation. So the take home from this was a pretty exciting one. It was actually one that we hadn't anticipated would be this extensive. We realized, and this is shown here in the dark black for insertions and dark red for deletions with respect to, at that time, GRC38, was that 92% of the insertions and 60% of the deletions, particularly in this size range, ranging from 30 to 5,000 base pairs, were novel, i.e. not detected by Illumina technology. So a big chunk of the genome, including parts of ALU insertions and L1 insertions, were not being sequence characterized or even identified by applying short read sequencing technology. So we estimated about 22,000 novel genetic variants, of which most of them were biologic, so they weren't artifacts of the cell line or anything to that effect. And that affected about 11 megabases of sequence. So just to put this in perspective, in terms of SNPs, this genome, because it was haploid, had just slightly around 3 million SNPs. So 11 megabases of structural variant base pair changes compared to 3 megabases of single nucleotide variation. So after Mark did that, we turned our attention to essentially characterizing the diploid genome. And this is work we did with the Human Genome Structural Variation Project. And here the concept was pretty simple. We want to be able to apply this to diploid genomes. They had 2 parental copies, one from mother and one from father. And the concept was simple. essentially convert the diploid genomes to 2 haploid genomes. And the way we did this was to use linking read technologies. So if some of you remember the technology 10x, also Hi-C could serve this purpose. In our particular case, we depended heavily on a technology developed out of the Netherlands called Stratified, which allowed us to essentially phase SNPs first from telomere to telomere. Once we had built this kind of phased SNP map, then to physically assign or partition the long reads either into haplotype 1 or haplotype 2, and actually build assemblies essentially directly from those specific sites. So we no longer were looking at a diploid rendition of the genome, but really 2 haploid resolved portions. And I'm not going to spend the time on this slide other than to show you that we actually compared 3 trios with essentially the long read. In this case, this was all continuous long read sequencing data on a Yoruban, a Puerto Rican, and a Han Chinese, which are shown here in blue, compared to essentially Illumina data from a Yoruban, a Puerto Rican, and a Han, the exact same individuals. And what you can see here is the gain that you get from applying long read sequencing, particularly with respect to insertions, and particularly with respect to insertions less than on the order of 5 kilobases in size. There's also a gain in terms of discovery and deletion, and just to keep in mind that this is a log scale. So there's actually a lot of additional real estate that's being recovered by essentially applying this technology. I want to highlight this little point here. We also applied bio-nano optical mapping technology, and there is in fact large forms of structural variation that live in this region of the genome that couldn't be resolved either with Illumina or PacBio at that time. The net sum of this was that when we had finished this detailed comparison of 3 individual genomes, we concluded that 73% of all structural variation was essentially detected as a result of long read application as opposed to short reads. So at the same time that we were doing these characterizations of the human genome in terms of structural variation, the data sets that we produced, both for CHM1 and CHM13, which are 2 hydatidiforms, were being used by Adam Philippi to develop new methods for sequence and assembly of genomes. And so this is a rendition of essentially GRC38, the one that most people still use as a reference. Some people still use GRC37. But the important point here is anything that you see with a black and a gray means that there's a gap or a transition. It's not continuous all the way through. Anywhere that you see essentially as white means it's a large region of a gap. So it's essentially no information across those areas. So at the end of 2019, using the CHM13 data, this is what the new genome looked like, applying essentially long read. Both at that time it was Oxford Nanopore primarily. And then with the application of Oxford Nanopore and Hi-Fi sequence, and this is pretty current, this is probably about a month ago, this is the current status of the human genome, at least what is called the telomere-to-telomere CHM13 genome. We would like to say 20 years after the Human Genome Project's papers that we've finished it completely. We're not quite there. You still notice that there are some gaps indicated here. But it probably it's between 5 to 10 megabases of the genome are unresolved. So most chromosomes are now, we're called working draft chromosomes because we're still finalizing this as part of the Telomere-to-telomere Consortium. But most chromosomes are now finished or in working draft finished state, where you have essentially sequence from one end to another. There was a publication earlier this year on the X, so I'm not going to talk about that. I'm going to talk about a recent release last month of the first autosome work largely of Glenys Logston in my lab, chromosome 8. So just a little note in terms of how we did chromosome 8. So the way we did it was a little bit different than the X. So what we did is actually generate 2 libraries. We had one from ultra long reads generated by Oxford Nanopore. And at the same time, we generated pretty high depth, over 30, close to 40x sequence coverage on that genome that we used for this study. So we had 2 different modalities of long read sequences. And the idea was pretty straightforward. The idea was that we would, specifically for complex regions that weren't resolved, what we would do is we would use the Hi-Fi sequence reads to identify unique k-mers. Once we identified those unique k-mers, years ago we called them SUNCs, or single unique k-mers that are uniquely identifying in the genome. We would then essentially barcode the ultra long reads that were generated. This would provide us a scaffold. And so this is showing you a scaffold across a centromeric region. And then once the scaffold had been created, remember the Oxford nanopore technology has higher error rates. So let's say 90 to 95% accurate in those regions. We would then replace it with the hi-fi assembled contigs that were present. So once we had the scaffold with the barcodes from the ultra long, we would then replace with the hi-fi contigs and have sequence that was 99.99% accurate in principle. So this is our short rendition at the top of chromosome 8. It's 146.3 megabases, and this is just showing you the telomeres. This is a half of a dot matrix, and you can see 1 to 2 kilobases of near perfect T2A triple G sequences with essentially degenerate T2A triple G sequences as you move in. Highlighted to the left here are the problematic regions of the genome, the ones that were kind of stubborn gaps. One some people might recognize is this one. This is the defensin locus. So this is an amazing region of the genome. It's one of the most common large inversion polymorphisms in the human population. It's about 4 megabases in size. The reference genome actually had the direct orientation present. The other thing that's remarkable, it's flanked by very large duplications. You can see them here, 500 to 600 kilobases in size. And when we sequence resolved CHM13, what we found was that there was, not only did we capture the inverted allele, but we captured another 644 kilobase duplication, which includes additional copies of the defensin B. There's about 3 additional genes that are added here. And then this is just read depth profiling showing Illumina data on several hundred human genomes to show you that the CHM13 copy is what we would expect kind of at the high end of this, while the human reference copy is essentially kind of mid-pack or a little bit lower in terms of the copy number of this 600 kilobases. We did other validations to confirm that this organization was correct. And so this is one of the largest alternate alleles. It was probably one of the most complex ones in the human genome. It was also a gap in the assembly. So what about the centromere? So this is the centromere, and this is the roughly 11 reads that were barcoded using hi-fi identified sunks across the region. We then filled it in with the hi-fi sequence. And so this is the structure depicted below. For those of you unfamiliar with essentially how centromeres are organized, particularly in humans, there's 171 base pair repeat that's repeated over and over again, but in terms of a higher order structure, so we often designate that as higher order. So this is going from the P arm. You can see there's often this divergent monomeric satellite sequence, which is not higher order. Then there's a higher order block that is, in this case, was slightly less than 2 megabases, and then going to the Q arm, a monomeric region interdigitated with other forms of repetitive sequence. Is it correct? Well, one of the things you can do is once you have the high quality sequence, you can digest in silico, predict the sizes, then do a southern digest, which is shown here on the right. This is a good old fashioned southern blot. And as far as at least the resolution you get from a pulse field gel southern, it's perfect. So we sequence resolved the structure of the first autosome using this approach. What did we learn? So this is the organization. This is work of Mitchell Folger in my group and Glennis Logston. What I'm depicting here is this heat map, but the heat map is essentially showing you in 5 kb blocks walking across the whole region, the sequence identity. So every piece of DNA is being compared to every other in discrete 5 kilobase blocks across. And so the percent identity is shown here at the bottom, going from these cold colors, which is low sequence identity, to high colors, which are things that are 100% identical. So I think you can appreciate right away that these sequences at the edges are very divergent. So they have 80% identical to essentially sequence to themselves, suggesting that they're more ancient. But interestingly, you also see that they are much more identical to one another than anything else. So there's no match to any of the other satellite sequences. Here's another layer. You can see this really at the periphery here. Kind of as you transition into the higher order, you see these tiny little blocks that are highly identical, but they're more identical to each other. And then you see this kind of block in the center, which is actually the most identical. And as you move out from this, things get more and more divergent, suggesting that we have both a mirror symmetry in terms of the organization of the centromere, in terms of the satellite sequences, and that is actually has evolved in layers, which was actually predicted years ago by George Smith and also followed up with Alexandrov, is that you would actually have this kind of layered organization of the centromeric satellites, with the oldest and most divergent being at the periphery. But the fact that you have these blocks, you can clearly indicate that there are distinct layers of the centromeric satellite. All right, so the last method I want to talk about before I talk about the actual kind of applications is really the attempt to actually go back to diploid genomes and actually fully phase and assemble them. So this is work of David Perupsky. It was a shared postdoc between myself and Tobias Marshall. He came to my group 2 years ago. And the idea was pretty straightforward. We would build on what we had learned from the structural variation project, take hi-fi PacBio data, using Fly or Peregrine or even Falcon. I think in the beginning, we built squashed assemblies of the genome. Use those squashed assemblies to identify the SNPs. And once we identified the SNPs, use the Hi-C, Strand-seq, or trio. So Hi-C is particularly work from trio, I should say, developing by Heng Li. Strand-seq was our own approach using essentially Strand-seq technology, but essentially phase the SNPs, in our case, telomere to telomere, as a result of this Strand-seq information. And then partition the reads, the hi-fi reads, into essentially haplotype 1 or haplotype 2. So these papers I believe they're both in bioRxiv. One, this is the one from Heng Li's group with respect to using Hi-C, and this is ours from David Perupsky and Strand-seq, using Strand-seq for that phasing information. So we applied it to essentially one trio. To be sure, we didn't use the parental information, so this is not trio phasing. This is trio-free and reference-free phasing, if you will. We focused on essentially the child, and we used the parental information to see if we got it right at the end of the day. So we generated 34-fold hi-fi sequence coverage from each of the 3 individuals. We generated 2.8-fold sequence coverage of Strand-seq data. We effectively generated 23 clusters where contigs were ordered and orientated. 95% of the SNPs were phased, and 81% of the CCS reads could be assigned to either haplotype 1 or 2. This is what the data looked like. We were able to assemble this Puerto Rican genome into essentially 2 haplotypes, accounting for about 6 gigabases, where the contig N50 was 25 to 28 megabases in size. And we used the parental information to determine whether we had assigned it correctly. So I'm showing you haplotype 1 and haplotype 2. And wherever you see a continuous blue color or a continuous yellow color, that means that we phased accurately. We got the right sequences on the right chromosome. Wherever you see a little discordancy, such as in chromosome 2 here, or you can see in chromosome 8, this is indicative of potential phasing error in this data. So we estimate that we got it right about 99.5% of the time for about 95% of the genome. And I say that because you can still see the Aerocentrics centromeres are not actually assembled here as part of this analysis. Once you have phased assemblies, you can then detect variants. So Peter O'Donnell developed essentially a phased assembly variant detection approach, which is just to compare the phased assembly back against the human reference genome, call all indels structural variants. And so this is your ALU insertions and your L1 insertions. And then project them. So this is now projecting them and showing you in a heat map where all the structural variation maps. You can see most of the genome is pretty quiet for structural variation, except for the ends of the chromosomes where we see an enrichment. And then this is the SNPs that came from that analysis. So we identified 4.1 million SNPs, 2.8 million roughly per haplotype, and the usual areas of divergence appear, such as the HLA region. The really exciting thing from this is that for much of the genome, we now have SNPs, indels, and structural variants fully phased, and we can do it without the use of trio information. So we think this is probably trio phasing, such as trio hi-fi ASM and so on, is probably still superior, at least based on the benchmarking that we have done. But this comes a close second in terms of what we've been able to do. And obviously not in all samples do you have the ability to have parental information. Importantly, if you care about loci, complex loci such as HLA, this is showing you that we actually assembled most of the HLA region, 6 megabases, in 2 distinct contigs. And obviously this is incredibly important for developing and optimizing immunotherapies for cancer. All right, so in my remaining minutes, I want to talk to you about the exciting stuff. We got the methods. We got methods that are working pretty well. I want to share with you some of the applications. So coming back to that centromere in chromosome 8, Glenys Loxton was particularly interested in understanding what she could learn about both the structure, function, and evolution of that chromosome 8. And one of the values of actually having the Oxford nanopore technology, as some of you know, is the ability to actually determine what's methylated and what isn't. So what's methylated is indicated here in red, and what's unmethylated is in blue. These are methylated Cs and unmethylated Cs. And as you expect, you transition into the centromeric region, it's hypermethylated on both sides, except there's this very conspicuous dip. This dip is about 76 kilobases in size. It is an area of hypomethylation in a sea of methylation. And what's interesting, back to the structure of the centromere, it's not the most highly identical homogeneous red portion that I showed you before. It represents essentially an area of diverse higher order that we identified kind of on the periphery, what we call layer 3 of this particular centromere. So one question is, does this represent the functional centromere, i.e., is it the point of kinetochore attachment? So once you have high quality sequence, remember that came from the Hi-Fi data, which was scaffolded with the ONT data, you can do things like ChIP-seq, and this is SEMP-A, which is replacing, essentially, as some of you know, the histone 3 that defines essentially the point of kinetochore attachment. And so this is ChIP-seq from 2 different cell lines for SEMP-A. You can see that it's a broader swath of over 200 kilobases, but the peak lines up, at least based on this level of resolution, almost perfectly with that dip, suggesting that this area of hypomethylation may be the point of attachment of the functional kinetochore. So if you can do a hydatidiform mole, can you do other organisms? So we actually investigated chromosome 8 specifically, generating Oxford nanopore ultra-long reads from a chimp as well as Hi-Fi data. And this is our first reconstruction of a centromere, probably the first that is done anywhere, of a chimpanzee going from the P arm to the Q arm for that chromosome 8 region. So once again, this is the heat map showing you it's clearly different than human, but there are a lot of similarities. You see the divergent monomerics in the periphery. You see this layered nature, actually more layered than human. You can see very distinct layers, blocks, if you will, of evolutionary relatedness. Again, with this idea of an assembly line moving the older stuff out to the periphery. So that's a chimpanzee, separated from humans 6 million years ago. Here's a orangutan, separated 12 to 14 million years ago. It's actually bigger. It's 4 megabases now. Once again, you can see this divergent pattern, very small monomerics in the periphery. But remember, this is 4, not 2 megabases of assembled sequence. You clearly see this layers again. So take a look at this, very large blocks of highly identical sequence, which is more related to itself than anywhere else. But interestingly, instead of actually having 2 domains in the center, or 1 domain, you see 2. So you see this sequence and this sequence block, or this layer, and they're actually very divergent with respect to themselves, suggesting to us, and this is just a hypothesis at this point, maybe they're fighting for dominance in terms of who will be the functional centromere on this chromosome 8 in the orangutan. And then last but not least, this is the macaque. It's a monster centromere. It's 10 megabases in size, sequenced and assembled. And one thing you should know about macaques, and it's been known for many years, they don't have higher order structure. They have a simple dimeric repeat unit that's repeated over and over again. But obviously there's this, again, this mirrored and layered nature again, where you can see this very highly identical sequences, but then this zone to the left and the right, which is actually very similar, just seems to be degenerate from that sequence. There is the divergent sequences on the periphery, but the other thing that you'll notice is that the repeat sequences, unlike humans and chimps and orangutans, which actually spans from 80% to 100%, almost all of the macaque sequences are virtually identical. So greater than 90% identical. So this is just a summary of, I think, the first orthologous sequencing, complete sequencing of the centromere, chromosome 8. And what you can do as well is, because you have the positional information on every 171 base pair repeat from alpha satellite, you can build a tree of relationships. And so what is shown here is a couple of interesting properties. Here are the higher order repeats that define the great apes, indicated along the top here. You can see that they're related to a specific subset of macaque dimeric and monomeric repeats. The layer 1, which is kind of at the periphery, is also related to this. But interestingly enough, you can see this very long branch here, indicative of what is the macaque dimeric and monomeric alpha satellite as well. but it's related to a very small portion, which we think represents the vestigial ancestral centromere located just on the Q arm of chromosome 8. So we think this is very cool because it suggests two trajectories for building centromeres, one that goes toward the production of higher order, the other, which was probably a false start in humans in the ape ancestral lineage, but actually became the predominant mode for building the macaque. So if you can do chimps, can you do multiple humans? So this is, I think, the first diploid centromere assembly. So this is work that Glennis has continued to do, looking at that Puerto Rican child, once again, reconstructing the configuration of haplotype 1 and haplotype 2. You can see they're very similar to one another, but there are differences. You can see that the size of the zone 5, layer 5 is different. It's larger in this particular haplotype. But it's a little bit surprising to us. We thought, based on old theory, that all the variation would be in the most highly identical sequences. But that does not seem to be a case. Look at zone 4. Zone 4 is actually quite large and is different in structure compared to haplotype 1 and haplotype 2. And even this zone, zone 3, seems to be varying among at least contemporary human populations. So once you build contiguous sequences, you can look at diversity. And this is just the beginning of that, but you can see that the diversity here is actually what you'd expect going into the paracentromeric region, one difference every 1,000 base pairs or 1,500 base pairs. But as you get to the actual higher order, there is an estimated increase of mutation rate at least about 4-fold in these areas of the genome. And interestingly, you can start to see areas that are under more constraint or potentially more constraint. And obviously, we will need more haplotypes reconstructed across chromosome 8 to address this. But the important point is we can now assess genetic diversity within humans' haplotypes because we can assemble these systematically. And then a point, just a plug for the actual T2T project. This is essentially the organization of what we now know about the Aerocentrics. So remember, this is a sequence that we largely has been opaque to our understanding for really almost a generation. There are still gaps, and that's where the remaining gaps remain, indicated here in white. But a surprising finding to us, we anticipated there would be rDNA, and in fact, most of the rDNA appears to be mapping to the gaps. But there's a very large proportion of segmental duplications that are present there. And so these are indicated by all these colored bars that you see. And what's also interesting is you can see that there are certain blocks that are shared among subsets of Aerocentrics, but not others, indicative that we now have the ability potentially to tag specific Aerocentrics versus others, which for those of you who don't know are highly related to one another, almost at the level of allelic variation. And perhaps the other surprising finding, and this is just a projection, we call it an Inca plot in our lab. This is showing you the relationships of the Aerocentrics to other regions of our genome with the duplication organization indicated along the periphery. We now know that about 29 megabases of the Aerocentrics is shared to non-Aerocentric chromosomes. So there's about a chromosome's worth, or at least a small chromosome's worth of DNA that's shared between Aerocentrics and non-Aerocentric chromosomes. So the last project I want to share with you is work of the Human Genome Structural Variation Project, a project led by Peter O'Donnell, Peter Abert, and Chi-Wei Xu. This is also work I do with Jan Corbel and Charles Lee. And it's essentially a goal to understand more or less the full spectrum of common structural variation. And obviously sequencing only a couple genomes won't get at that question. And so this was an effort to sequence, we made a decision about 1.5 years ago to sequence 32 reference genomes, kind of indexed from essentially the 1,000 Genomes project, so selecting an individual from each of the populations, and then applying our phased and assembly-based approach to sequence resolve these genomes, build new references, and characterize all forms of genetic variation with an emphasis on structural variants. So we applied the method developed by Peter Abert and David Perubsky. It's not, they call it now PGAS, phased genome assembly using strand-seq. So it's a simple idea. We did it with both CLR as hi-fi, which is generate squashed assemblies. From those squashed assemblies, identify the SNPs. Then phase the SNPs using essentially the hi-fi data, or I should say the strand-seq data, and then assign the reads, about 80 to 85% of the reads, either to haplotype 1 or haplotype 2, assemble genome, call variation, fully integrated with SNPs and indels. So this is work over the last month, which was a real tour de force. So 32 genomes generate 64 phased assembled human genomes, essentially. And what I'm showing you here on the bottom in red are the variants that every phased human genome has, but the human reference does not. So there's about 1,200 of these. These are likely errors in the human reference. Most of them are outside of genic regions, but there are some that are in regulatory and genic regions. And then this purple line represents major allele. So the allele frequency is greater than 50%. So this is what most humans have. And you can see that this is plateaued out pretty early on. So we've got all of that more or less figured out. This is the polymorphic. So this would be above 1 to 2% allele frequency based on a number of genomes that we analyzed. And you can see that it's almost asymptotic. And adding only a few hundred new polymorphic sites for every new haplotype that's generated. However, and this is just the singleton counts, if you look at the Africans, and we broke out specifically the non-African versus the African, as you might expect, there's more genetic diversity in Africans. And so Africans are adding another 1,000 structural variants for every new African genome that we characterize. So going forward, and we will do more African genomes to capture more of the common variation that's being missed from the human population. So once you have, and so from this analysis, we identified 107,000 sequence resolved structural variation events. And with a tool that I'm sure you'll hear more about today or tomorrow from Tobias Marshall's lab, Pangenome, we have the ability to go back to Illumina data. And this is data from the 3,200 samples from the 1,000 Genomes project that are now been sequenced at 30-fold sequence coverage with Illumina. We can now genotype a subset of these highly accurately. What did we learn? Well, something that we already saw last year, but we confirmed. About 30% of all structural variation maps to the last 5 megabases of human chromosomes. That's just a statement of fact now. We've seen this over and over again. This is particularly true for VNTRs and particularly insertions. And the reason we think it is that because there's an excess of double-strand breaks allowing for such structural variant events to occur there. So it's a 4 to 5-fold enrichment of structural variation at the ends of chromosomes. The other thing that we can do is we can go back and generate RNA-seq data, and this is work that was done by Mark Bonder and Arvis Sulivari from my lab, is to look for new expressed quantitative trait loci that may have been missed if people were depending only on SNPs. So when we did this analysis, we have 57,000 indel eQTLs, and 2,100 SV eQTLs at a false discovery rate of 5%, for which 713 of the lead indels are, or the lead variant, this is compared to SNPs, is the indel, and 34 where the SV is the lead. So this is just an example of a gene, a mitochondrial G2PAs, in which there's an 89 base pair insertion, which is the lead eQTL associated with, in this case, decreased expression. And this is a lipase gene. This is a gene in which we have a deletion. And the deletion is about 1,000 base pairs located right at the end of the gene, but not infecting the gene. And this is associated with essentially increased expression of lipase. This is a particularly interesting one because there's a GWAS signal, which seems to be associated as well with essentially heart failure and reduced ejection flow and heart rate over this region. One thing I'm particularly interested in is essentially stratified variants. And the reason for that is we're looking for variants that have risen to high allele frequency in the human population or specific human populations that may be showing signs of adaptation. So not all structural variants are bad. We know that some structural variants actually confer benefit, so this is an attempt to identify them. So this is work that couldn't be done in the 1,000 Genomes project, but now that we have sequence resolved the structural variants and we can genotype them, shown here are the genes and structural variants in terms of the size, where we actually identify within 5 kilobases of the gene, either internally or either 5' or 3', a structural variant that seems to be highly stratified. So the measurement here is max PBS, which is a branch-specific estimator of stratification. So you might recognize one. This gene here is LCT. It's also known as lactase. It is one that's actually, in this case, it's actually a deletion in the European population. So the blue is indicative of the European. And the reason that it got our interest right away is lactase, as you might know, is a classic human genetic trait where lactase persistence is associated with the ability to consume milk essentially for past the age of 2 or 3, which is correlated with 3 or 4 SNPs that are thought to play an important role in allowing for that persistence of lactase expression. So we have essentially a 3.9 kilobase deletion in Europeans. So in other words, in Africans, it's a 3.9 kilobase insertion. It's in complete LD with the SNPs that are already known to be associated with lactase persistence. So the deletion, I should say, is in complete LD. And so we're not making the claim yet. It could be just a simple case of linkage disequilibrium, but we think it's interesting that this 3.9 kilobase piece of DNA also has transcription factor binding sites, close to about a dozen of them that may be actually targets for controlling the regulation and expression of lactase. So this is something worthy of follow-up, we think. So the last points I'll make is that in addition to essentially characterizing structural variants that are essentially stratified in the population, we have the ability to identify structural variants that have evolved specifically and are shared among all humans. So using long-read sequences published a few years ago, we characterized what we think is the largest genetic change between Neanderthals and humans. It's 105,000 base pair duplication that all humans have. We estimated at 250,000 years ago. It's particularly interesting because this piece of DNA increases the copy number of a gene family called BOLA2. BOLA2 is important for iron metabolism, and all humans have extra copies and extra expression of this gene as a result of this duplication. The bad news, as some of you may be familiar with this, is that this duplication creates a susceptibility to the second most common cause of autism in the human species. So it's a bit of a yin for your yang. You get additional copies of genes, but at the same time, you're predisposing to a very common cause of autism, the 16p11.2 deletion. So last year, we had the ability to follow up on this locus in more detail. And one of my postdocs, Bench and Shea, revisited some old data, which is in the exact same region of the genome, but a completely independent duplication that he identified specifically as found in individuals of oceanic descent. These are Melanesians. They have an extra copy. They're either heterozygous or they have a complete, and both alleles, they have 2 copies. This is indicated by the 4 here. And what's remarkable here is that no other human population that we surveyed, at least on the planet, has this duplication. Except there is one individual. This individual up here, this DNS, represents Denisova. So some of you may know Denisova is like the Neanderthal. It's an archaic human that lived 50,000 years ago. And it has a unique signature is that it actually, although Neanderthal lives in most Asians and Europeans, at least parts of its genome, this particular species only introgressed with Melanesians, or at least the ancestral Melanesians. So we genotyped a larger pool of them, of Melanesians. So this is work from Jonathan Friedman with PCR assays we designed. And what we determined is that the allele frequency of this very large duplication is 80% allele frequency in Melanesians and 0 in most other continental humans. So using, in this case, we use CLR data. We reconstructed the sequence. It was much bigger than 100 kilobases. It was a 383 kilobase insertion, mapping to the same region where we have the Homo sapiens specific duplication that evolved 250,000 years ago. This 383 kilobase duplication is specific to the Melanesians. We estimate based on divergence that it arose about 300,000 years ago. We believe it arose in the Denisova archaics, and then 50,000 years ago was introgressed into essentially the Melanesian ancestral population. It then rose to high frequency, and the data suggests as a result of adaptive or positive selection. This new piece of DNA carries at least 2 duplicate genes, and I'm showing you one of the genes right here. We called it MPIP-B16. It shows evidence of an excessive amount of amino acid replacement changes, as well as signatures of adaptive evolution consistent with a selective sweep or partial selective sweep in the population. So this is cool, right? Because this is actually discovering entirely new swaths of DNA that encode genes that are stratified in populations that show essentially not only structural variation, but signatures of adaptive evolution. We don't know what the actual trait is. It may be an ancestral trait that no longer is important, but we know now it is present in almost all Melanesians in the world. This is just one of many that we've now identified. And so the really exciting thing, if you stop and you think about it, the fact that we're building these new reference genomes from individuals from Europe and Asia and Melanesia will allow us in principle to reconstruct ancestral structural variants, because 30% of Neanderthals lives in us today as a result of introgression events that happened 30 to 50,000 years ago. And so it's, I think, a fairly unique application of the long-read sequencing is to reconstruct the full structure and potential signatures of evolution in these species that no longer exist other than through us. So in summary, I hope I've convinced you that long-read sequencing is revolutionizing our field. It is allowing us to really get a handle on really an underserved and understudied form of genetic variation, which essentially contributes more variant bases than any other form. Combining both HiFi and Oxford nanopore technology, we think is the solution for generating telomere-to-telomere assemblies. And I hope I convinced you that we've been able to go where no man has gone before and sequence resolve very complex things such as centromeres. And then finally, combining HiFi sequence data really with linking read technology. And in our case, we use StrandSeq, but HiC is another option. We can now phase and assemble about 6 gigabases for every genome we analyze. So we're getting 2 genomes for the price of 1. But the really important thing is that SNVs, single nucleotide variants, indels, and structural variants are fully phased. So if you stop and you think about it, this is essentially what we want to do clinically. This is what we want to do going forward for human genomes. And while I know that the clinical world is slow to adopt whole genome sequencing, period, I think ultimately this is the way patients, children born with developmental disabilities, essentially our care will be generated by essentially generating long-read phased human genomes where all genetic variation is understood. I don't have time to go into this, but I'll just mention the Human Pangenome Reference Consortium. So building on these ideas of converging both on this idea of actually telomere-to-telomere assembly as well as human diversity. This is a project being led out of the UCSC, which I'm a co-PI as well, to actually characterize 350 human genomes that better represent the diversity, to build better references, to build a pangenome that will improve genotyping and associations in the future. So these are all the folks that contributed to this work, at least a partial list. I should add from PacBio, a lot of the initial contributions here came from encouragement really with Mike Hunkapiller, which I didn't have on the list here. I should have him on the list. I want to acknowledge particularly Glennis Logston, David Perubsky, Mitchell Folger, and Benson Shea for allowing me to share with you unpublished work. Also my colleagues Adam Philippi and Karen Miga from the T2T and the HGSVC, which is the Human Genome Structural Variation Consortium, both Tobias Marshall and his postdoc Peter Abert. And this is a picture of how my lab now interacts. Notice all the smiley faces. You have to trust me that there's still smiles even though when we have to do this, which we do this far too often, underneath those masks. But without those folks, we couldn't do this work over here. So with that, I guess I want to welcome Jonas back onto the quote virtual stage. If there's any questions, if we have time to take them, I'd be happy to. So I'm going to stop sharing now.
Great. Yeah, thank you very much, Evan, for this amazing talk. We do have a few questions and we have a little bit of time. First one is amazing work. In these highly variable regions like the Fenton, how well marked are they by haplotypes/SNPs, i.e., how well can common SNPs predict the structure variation?
Yeah, that's a great question. So we haven't looked specifically at that. The sequence is relatively new to us. I can tell you that for a lot of, so let me just back up, that in terms of structural variation, there are 2 types of structural variants simplistically. Things that we classify as binary, so either you've gained or you've lost a sequence. And those tend to actually genotype very well with flanking SNPs. Then there are things that are often you see in the literature called multiallelic CNVs or multi-copy CNVs. And these are things where you are 2, 3, 4, 5, 6, like the defensins. And historically, and this goes back to literature almost more than a decade ago, these genotype very poorly by flanking LDs. So basically SNPs that are located adjacent. And the idea behind this is that the frequency of recurrent mutations is so high that you're getting identity by state as opposed to identity by descent for a copy number 3. But that's a case-by-case study, right? So we haven't done it specifically for the defensins, but it's a really good idea because if you can genotype, so one thing that we haven't been able to genotype, but we now can, is genotype within the duplications. Because those same sunks, right, those single unique k-mers that allow us to uniquely assign and assemble the genome over those regions are the same things will allow us to genotype them. And I think that's probably the way to go forward on them, is have multiple sunks go in and genotype. And it won't be perfect, but it will actually be, I think, better than taking flanking ones.
But I mean, for what you showed, for example, defense, I mean, it was so structurally polymorphic, and you're running out of SNP variation to characterize all of that with flanking SNPs, right?
Yeah, it depends upon the evolutionary age, what we call the coalescent, right, for these different structures, right? So that's also a reason why we need 350, human genomes, right? Because we need to sample enough of the diversity to understand what the tree looks like, not between apes and or non-human apes and humans, but actually among humans. Once we have that tree topology, we can do very intelligent and strategic genotyping in Illumina data.
Yeah. The next question is with regard to validation of the centromere. So the question is, nowadays, it is rather common with HiFi to get, with the assemblers to go through plant centromeres with HiFi reads. What would you recommend to validate them bioinformatically or experimentally?
Yeah, that's great. It's been a painful thing. We've solicited a lot of help from others. So, for example, the pulse field gels, there's relatively few people that can do this well. We're not one of them. So we had a colleague from NIH who helped us on those particular gels. There's also there's qPCR-based methods. To be honest, one of the things that I found very helpful and useful for validating is to redo the experiment. So when we built the CHM13 assembly, the first thing we did is we also compared it against our CHM1 assembly of that area. And you'll automatically see differences, and some of those are biologic, right? You hope most of them are biologic. But then you can test specifically for those structural differences between haplotype 1 and haplotype 2. Those are much easier things to do than to try to reconstruct or do pulse field gels for every centromere that you've characterized. Yeah. But yeah, I mean, it's a tough one. The same goes for signal duplications. You get these configurations that you've never seen before. One of the things that we did is pull out backs. We still do this. Pull out large insert backs from old libraries, sequence them, and then compare them and see if the structures are the same. I'm happy to report that we're doing this less and less often. And that's largely because it's easier to generate ONT data or more HiFi data on the same sample than to do the laborious work of pulling out large insert clones and sequencing them.
Yeah. Next question is about the stability of these centromere structures that you see with regard to being passed on to the next generation. If you have some tree information, if you have them resolved.
Yeah, that's something that we really want to do. So we haven't done it yet, right? So we just, like I said, we just built the first diploid sample. And the way to do this, to be honest, is not to use cell lines. The Karen Miga has reported several examples where she's seen artifacts that we think are clearly changes that are occurring somatically, quote unquote, in the cell lines. So the way to do this is to go back to blood and actually get blood DNA from a mother and a father and a child. That's the way to do it. So for questions of de novo mutation, which I think would be absolutely fascinating, that's exactly what we're planning on doing at this point.
Another question is regarding cancer. Do you think it will be possible to resolve these chromosomes with the centromeres and potential centromere duplications in cancer genomes and other centromere-based rearrangement in cancer?
I get scared when I think of cancer genomes because of the complexity that goes on there. The big problem there, we've looked at this a little bit with our strand-seq-based methods. The trouble with that is the ploidy problem. So we're good at 2. And so for those of you unfamiliar with strand-seq, there you're basically, you take a cell, you allow it to go through one passage, you BRDU label, you incorporate, then you destroy the newly synthesized strand, leaving either the Watson or the Crick strand left on each of the homologs, and then you have directed sequencing. That's what gives you that information. When you get 3s and 4s, it all goes to hell in the handbasket, right? Because you don't actually have, there's too many Watts and too many Cricks floating around, right, to make this unambiguous. So our method is really dependent upon counts of 2, right, for the phasing. But I can imagine with the high-C based approaches that this might be possible. You still have the problem is that those, copy 3s and copy 4s are coming from some other homolog that's virtually identical, right? So disentangling that would be a challenge, I would imagine. But, maybe the approach there is, screw the ancillary technologies and just do Oxford Nanopore plus I5 and use scaffolding from the ONT data, the ultra long data. My biggest frustration with that is that it takes us a long time to generate ultra long ONT data, because you have to prepare very large, high molecular weight fractions of DNA, and so the yield is for us at least, and we typically use the GridION system because we PromethION generates kind of standard libraries, but we really want the ultra long data for this purpose. And so if there was a way to get that procedure, increase in throughput, I think it would be very complementary to the strength of the hi-fi data, which is the accuracy.
Yeah.
Plus, you get the bonus of the methylation status, which is also very important for cancers, right?
Well, as you know, I would like to encourage you to look at the methylation with the PacBio data as well, because it's there also, the kinetics of the polymerase information.
We looked at this, 4 years ago, we had a student from Japan that came specifically, worked for 6 months in our lab, but for us, the coverage required was too high to.
Right, and it's still there in the hi-fi data now, and you get the intramolecular consensus just like you get on the base calling. You get it with the methylation, with the kinetic signal as well. So I'm thinking that might be a nice way to cross-validate the methylation signals, that because you have 2 independent technologies and you can compare them against each other.
Yeah. Well, always certainly willing to try again.
Great, excellent. Well, thank you so much again for this terrific presentation, and thank you everyone for attending and for the questions, and a virtual applause, but really great talk. Thanks so much, Evan.
Yeah, my pleasure, Jonas. We'll see you again, hopefully sometime in person.
That's right. I hope so too.